Yeah, I have one over there. Yeah, so however you want to do that. Started here, I know there probably have a few more people coming in, but I'm really glad to welcome you to the College of Life Sciences seminar today. I'm Brent Nielsen in the Microbiology and Molecular Biology Department, and we're really glad today to have Dr. Henry Daniell, who's visiting from University of Central Florida, to present to us. Dr. Daniell is the Pegasus Professor and Trustee Chair at the University of Central Florida in the College of Medicine in the Department of Molecular Biology and Microbiology there. And I've known Henry for quite some time. We actually were on the faculty together at Auburn University in Alabama for a few years. And then he moved to Florida a year or two before I came here to BYU. So Dr. Daniell got his PhD in India. He then came to the United States and did postdoc at University of Illinois Urbana-Champaign. And then he was on the faculty at Washington State and University of Idaho jointly for some time before he went to Auburn, and then of course now in Florida. He's published very widely in a wide range of chloroplast molecular biology, but for a good time now has been working on chloroplast genetic engineering and plant development of interesting compounds, and basically what's in his title, the plant-derived pharmaceuticals, vaccines, and biofuels. So he's published very widely in PNAS, Nature Biotechnology, many other top journals in these areas. So we're very glad that he can be here, and we'll turn the time over to him now, and just to help me welcome him here.
Thank you, Brent. It's really a pleasure to see Brent again. We have known each other almost 30 years. I met Brent at UC Irvine when he was a postdoc fellow, and I was at Washington State University in Idaho at that time. And then we published a PNAS paper at that time, which lays the foundation for the work that I'm going to be presenting today. And then again, our paths crossed, and I forever should be grateful to Brent for inviting me over to Auburn. And because of Brent, Brent offered me a job, and then it eventually turned out to be a tenure track, and then eventually I became a professor at Auburn. And then, so thanks, Brent, for all the wonderful friendship that we have had, and the impact of your friendship on my career. Today, the focus of my talk is going to be, I see a lot of students here, undergraduate, graduate students, the importance of interdisciplinary science, how science, your future is not knowing and being comfortable with one specialized area, but having the skills in a number of different areas, and then have a global solution for challenges in health and food and environment. So today I'm going to give you several examples, giving the concepts and also the advantages. If you look at the logo, you see the leaf there, but then it's overlaid on the logo for health. That's our medical school logo. So just two totally different fields. You don't think of science and health science and agriculture, but yet that's what I'm going to be presenting to you to give you an idea of the importance of interdisciplinary science. There's a great need in health for developing cures for diseases, vaccines for infectious diseases, and so on. And more recently, after September 11, we have heard a lot about anthrax and plague and so on, and vaccines for bioterrorism agents. And I'm sure you are participating in the healthcare debate. The past few years, we have been spending 150 or 170 billion dollars on just one disease, on diabetes. So, how is it treated? By injecting insulin, insulin-dependent diabetes, but can that be cured? If that is so, then this whole $150 billion industry is an unnecessary industry. So I'm going to give you some significant examples of those and scientific concepts and proofs. And if you look at the current versus the future directions, all the complex machinery that you see is a fermentation system. That's how all of our insulins, interferon for cancer treatment, all of the cell culture systems, either it is yeast or E. coli or CHO cells, Chinese hamster ovarian cells, all these are used. And they all basically use the same fermentation device. And then they need to be purified, and once you purify, then it's 99.9% pure. You can't have any of the host proteins in that. And then refrigerated, cold transportation, sterile injection, and all of that. So the future technology that I'm going to present today is instead of doing this complex, expensive process, just express these proteins in orally deliverable systems like lettuce and make capsules and orally deliver. So you're talking of pennies, because you can't sell powdered leaf lettuce for example, interferon right now is in Orlando, $40,000 treatment for 3 months. This is for hepatitis C or for cancer treatment. So the how is this possible, and how this and how valid this is, and all these aspects that I'm planning to present today. To express foreign protein in a plant cell, then there are three choices. You have the nucleus, the chloroplast genome, and the mitochondrial genome. And almost 25 years ago, I proposed that this could be done through the chloroplast genome. Again, this concept was the candidacy proposal that I wrote when I was a PhD student. So your thought process as a student will go a long way in advancing your career. And the rationale behind this is that the genetically modified crops were introduced in the mid-'80s and in the real world in the 90s. And now, if you know, your soybean is almost 97% of soybean grown in the US is genetically modified. So Europeans have some concerns. One concern is that the genes are flying out through pollen. So I was proposing to address this by introducing genes through the chloroplast genome, because as you know, chloroplast and mitochondrial genomes are maternally inherited. Therefore, you could overcome this concern. And when I was in Pullman, I mentioned Brent's role in my career. There was another important person whom I should thank is Lawrence Bogarat. In invited me over to Harvard when I was in Pullman, and at that time, the gene gun was invented at Cornell. So I was driving back and forth, and eventually we used the recombinant DNA vectors made for chloroplasts and then shoot them into plant cells and do selection, which is very specific for chloroplasts, so that ultimately you have all of the chloroplast genomes transformed. In fact, the first gene gun that was built in John Sanford's garage at Cornell was used the .22 caliber bullet from Kmart. And we used to have stacks of bullets in our labs, but nowadays we are more refined and we use a helium gun. And it's scary because we have stacks of bullets and we will have the Bunsen burner, side by side. So it wasn't really fun to have this. So for oral delivery system, then you can see now we have developed this system for lettuce. You can see how a single cell, and this is something very unique about plant cells, a single cell can become a whole plant. This is called pluripotency. For humans, we have pluripotency of stem cells. A cell type can change into another cell type, but the stem cell cannot become a whole organism. Plants can do this, and this is called as pluripotency. So that first cell that starts dividing, all the other cells that didn't capture the gene will be killed, and then it multiplies itself, and then ultimately we take it to the greenhouse and get seeds out of that. So once you get that, then in my lab, early we start with recombinant DNA, we do the gene expression in E. coli, then we do the gene expression in plants, tissue culture. Before, of course, we start making the vectors, we also sequence the genomes and isolate the genes, so one has to be familiar with genomics and bioinformatics. And ultimately deliver it. If you have the capsules, then you don't have to purify the protein. But if you're injecting, then you have to purify the protein. So you learn all the protein purification techniques and then compare injectable versus oral delivery and ultimately see the outcome. Those are all the results that I'm going to present. But as you can see, the freeze-dried leaves can be stored at room temperature. We have stored them for years with no degradation or significant degradation in the antigens. You can eliminate cold storage, transpiration, and also needles and other aspects. But science gets complicated once you develop a hypothesis. In this case, then we have to, first when we orally deliver it, then we have to make sure that we can deliver this protein into the circulatory system in animals. How do we do this? Because if all the proteins that we eat end up in our blood system, then we can't survive, right? So there's a strong blockage of our proteins getting into the system. So one way we accomplish this is once you express a protein, in this case you see a green fluorescent protein expressed, and then once we express this protein, we figure out the receptors in the gut epithelium where GM1 is a common receptor, and then we found like cholera, how does it inject its toxin? It produces this protein, transmucosal carrier protein, that binds to GM1 and then injects the toxin into the circulatory system. So we took out that and then fused to that protein, green fluorescent protein, and then when we orally deliver, you can see glowing liver, glowing ileum. So we figured out a way to cross the barrier and then deliver this protein. Once you deliver this protein, then you can ask the question, can you really test vaccines on biopharmaceuticals, and what are all the possible challenges in doing that and advantages in doing that? We have worked with both the anthrax and plague. If you go to CDC, in the bioterrorism list, anthrax is number 1, plague is number 2. We still don't have a vaccine for plague. We have one vaccine for anthrax, which the military people are refusing to take because it contains the anthrax toxin. This is the old 50-year-old technology that's used to produce it. where you grow the Bacillus anthracis and harvest the vaccine protein from the culture filtrate, so which gets contaminated with the toxin. So here we clone the vaccine antigen gene, express this in the plant, and then when we orally deliver this, you can see the oral delivery, all of the animals survive, whereas injectable, only 1/4 of them survive. And I'll give you the immunology background as we go along. And the reason for this heavy challenge is because we cannot have anthrax and plague in university lab. So we ship animals to the Army lab, and these guys were quite eager to test our animals, so they challenged them with 50 billion spores. So if they challenged 50 billion spores, the injectable vaccine wasn't as effective. So if you can see the table here, the spleen of the animals that died had 10 billion spores, whereas not a single spore in the orally delivered animals. So oral delivery system works better than the injectable system, and I'm going to give you additional immunological evidence as we go towards that. And this paper just came out on malaria and cholera vaccine. NIH has put this paper on their website if you go to NIH. So currently there is no vaccine for malaria. There's one vaccine for cholera, which is not very effective. And even last year, I think in Vietnam, they had floods and a lot of people died in cholera. This is the one among the top 3 diseases listed by the World Health Organization. And the immunity of the current vaccine, which is very expensive, there's only one vaccine right now, it's lost in children. So we use our approach. Again, here we compared the diarrhea after challenge with the cholera toxin. And as you can see, oral still worked better than the injectable when we challenged it with the cholera toxin. And why is this? Because if you, in this case, if you read this paper, you will see that we examine all of the immunoglobulins in immunized animals. And then we found that 2 immunoglobulins play a prominent role in confidence protection. One is the IgG1, the other one is IgA. When you inject it, only the systemic immunity, or you develop IgG1, but if you orally deliver it, you develop both IgG1 and IgA. And IgA confers mucosal immunity. So for mucosal pathogens, then this dual protection is quite powerful. And that's why every time we do this oral delivery, we find fascinating results. And so in this case, we examine all of the immunoglobulins, and these are the 2 key things that we observed. So I said malaria, there is no vaccine. It almost infects half of the global population, 350 to 500 million infections each year. Here also, this vaccine we developed was dual, cholera as well as malaria. So here we collected the sera and then did these immunofluorescence assays against the, we used dual antigens here, 2 different antigens for the malaria. As you can see, it binds to the malarial parasite, and we did the assays. We used the standard from NIH. Both oral and injectable groups had greater than 100% kill on the malarial parasite. So we are then able to develop a dual vaccine for malaria and cholera using this chloroplast transferase system. I don't have time to cover a lot of other diseases, but if you go into the literature, you will see a whole lot of other vaccines that we have developed and a few other labs have developed using this concept. But I'm going to take you to one other important aspect of the principles of immunology, which takes us to understanding diabetes. Insulin-dependent diabetes is an autoimmune disorder in which the body's immune system attacks the pancreatic beta cells that produce insulin. So ultimately, to stop this immune attack, the beta cells stop producing insulin. So the current treatment then is to inject insulin, that recombinant insulin, produced in yeast or E. coli. But we looked at it very differently. Instead of just producing insulin and injecting insulin, why don't we stop the immune attack? And so to do that, then the basic concept here is then present the antigen to the gut immune system. And the gut immune system, if it rectifies this disorder, then once the immune attack is stopped, the beta cells may have the ability to regenerate, and the pancreatic beta cells, if they regenerate, then they will produce insulin. So you don't have to inject insulin. So this is what I'm going to walk you through. So here you see the panel where we have 3 different negative controls, and this is the pancreas that regenerated. And as you can see how bad the pancreas is damaged in diabetic mice. And this has a very intact structure. So we scored this, it's called insulitis score, and then we almost got a perfect score of insulitis after 6 to 8 weeks of oral delivery of human proinsulin, which is expressed in chloroplast. And in this case, we did this with the lettuce cells. And then we wanted to further confirm production of insulin, and we immunostained insulin. The green color that you see is a stain for insulin. And you can see how the pancreatic structure is disorganized, whereas this is intact. And you can see, and we also look for the cell death marker caspase 3. You can see that there is no cell death in this case, and it's recovered, and therefore the structure is intact, and they're producing insulin. And does it translate into blood sugar and urine sugar levels? Yes, it does. So we measured the urine sugar levels and blood sugar levels. These animals, the diabetic mice, usually die in 13 to 15 weeks. In this case, after oral delivery, they were never given insulin. They had their full lifespan of 50 weeks. That's their entire lifespan. So by fixing the immune disorder and not giving any more insulin, we were able to use the mucosal immune system for development of what's called as oral tolerance. And to check the pathway, in what pathway this fixed this, we looked at the MS2 pathway. In Th2 pathway, then you look at both the IL-4 and IL-10. You can see the production of IL-4 and IL-10, confirming the hypothesis that the immune system disorder has been fixed. And therefore, they are able to reorganize the pancreas and stimulate this. So this basically, juvenile diabetes starts in children, and if we give this lettuce capsules early before the onset of diabetes, which can be monitored by looking at the C-peptide levels, then the onset of diabetes could be delayed. But in more advanced cases, in adults where they have had diabetes for several years, decades, then there are other proteins that act as autoantigens. GAD is one of those. So we could use GAD and then fix after the onset. And there are proteins like PDX1 that stimulates regeneration of beta cells in the pancreas. So you can have a cocktail of proteins depending on what stage the disease is. And so this is a pretty different concept. Genentech developed insulin, producing insulin in E. coli. Novo Nordisk sells insulin produced in yeast. Over $25 billion insulin is sold. Last year, just the blood test strips were sold for $9.8 billion. So when we talk of healthcare, the real problem is also not only political solution, but also technological solution. And this would be one example how science could help solve the health crisis. Even more important is that when Genentech made this, they knew how insulin, let me back up and say, insulin has 3 peptides, A, B, and C. Genentech knew how to make only A and B. The technology was developed in the mid-70s, and that's the product that was approved. Till today, that's what's being sold. So people who get insulin for years, all these past 30, 40 years, do not get C-peptide. Absence of C-peptide, prolonged absence, results in neuropathy, nephropathy, damage to the retina, all of these things. So here, we show that when you regenerate the pancreatic cells, it produces also the C-peptide. C-peptide levels goes down with age, but even in old mice, you can see the C-peptide levels are coming up back. Therefore, when we do human clinical trials, even if they are taking insulin, we can monitor C-peptide levels to see whether this autoimmune disorder is being fixed. So this is a very important marker for clinical trials to find a cure for diabetes rather than just giving insulin. And we have a lot of work cut out for us. It's not only diabetes, you can see diabetes among autoimmune disorders at the bottom, thyroid, multiple sclerosis, hemophilia, a lot of other diseases that could be cured using this concept. So, not satisfied with what we got in diabetes, I was very eager to quickly move into other autoimmune diseases. And we have a paper coming out in PNAS probably in the next 2 to 3 weeks, where we took this to other genetic disorder, hemophilia, where the blood clotting factors are not produced, just as the insulin is not produced here. The challenge there is that when people receive blood clotting factors, the body develops antibody, and then some of that antibody, IgG, are converted to IgEs, which is responsible for allergy and anaphylactic shock. So this, we did a very large animal study with 25 control and 75 treated. Not a single treated one developed IgE, whereas all of the control animals, when they get this shot of blood clotting factor, they just go around the cage and then die. So we rescued these animals by giving antihistamines, and then we collected blood and then found out that the IgE levels was growing through the roof, and within minutes they have anaphylaxis and then they die. So we have now established this concept in other autoimmune disorders or other genetic disorders where this kind of immune fixing the disease by oral, this is called as oral tolerance, and how this could be a pretty powerful approach, and how plant cells can be used as oral delivery system. Because remember, it's important to protect this antigen from the acids in the stomach and enzymes in the stomach. So this bio-encapsulated cell wall protects them, and then it needs to be released in the gut. And so there are a lot of other vaccine antigens and biopharmaceuticals have been expressed in the chloroplast. And so I'm at this point, I want to also bring to your attention that this platform can be used to fix other environmental concerns as well. One of them is biofuels. But before I get into that, maybe I'll stop here and then take some questions on the immunological aspects of it and also any other aspect of this. Yes?
So you're administering your vaccines through cholera toxin primarily? Is that your main delivery?
Yeah, it's unfortunately the literature named it toxin, but it's a non-toxin. It's a non-toxic subunit called B subunit, beta subunit.
The 5 beta subunits still deliver your vaccine as if they were an A subunit, do they?
Well, there is no A subunit we use here. We take the B subunit, we fuse the whatever protein that we want to deliver, and this B subunit, inside the chloroplast, they form a pentameric structure, form 5 disulfide bonds. And when it is in the pentameric structure, then it can bind to the GM1 receptor and then helps in transmucosal delivery.
So are your vaccines embedded in the B subunits, or are they a separate subunit?
That's not always necessary. For example, our F1B vaccine antigen that we delivered for plague did not have CTB. Yeah, some of them we use and some of them we don't. And we are working on several other transmucosal carrier proteins. One of them is the protein transcription domain that HIV uses to move from cell to cell. And so we are trying that. There are also other peptides that take the protein straight to dendritic cells called the DC peptides. So we have a bunch of other proteins that we can use to achieve the transmucosal delivery. Yes?
You talked about with IgG and IgA levels. With the injectable vaccine, you only had IgG levels being increased?
IgG1, yeah.
IgG1.
Mm-hmm.
I'm curious, how did the IgG1 levels compare between the oral vaccine versus the injectable?
Good question. Actually, the IgA is derived from IgG1.
Mm-hmm.
So you initially have the IgG1, and then ultimately you derive IgA from that. In plague, we got even higher IgG1 levels with oral delivery than with injectable delivery. But oftentimes the IgG1 level will decrease as the IgA level increases. Yes.
The diabetes treatment went a little over my head. What's the antigen you're delivering? And repeat to us the effect that has on the autoimmune response going on in the pancreas.
Okay. The antigen is a human proinsulin, which contains A, B, and C peptides. So we clone the gene and then we express it in the chloroplast. And the basic understanding is that first the immune system attacks insulin, and then it attacks the cells that produce insulin, which is the beta cells. So this is where the autoimmune aspect of this. So if you use the mucosal immune system by presenting this to the dendritic cells, teach the immune system, the body constantly looks at what we eat, and then they compare, is this protein toxin, is this protein pathogenic, should we make antibodies? So these decisions are made by the immune system every single day. So when they compare it, they find that, well, there is a comparison here. So then at that point, the thymus 1, which is an inflammatory pathway, switch to thymus 2. The indication for that is upregulation of IL-4 and IL-10. And once this IL-4 and IL-10 establishes, then the pancreatic beta cells start regenerating. And then when they regenerate, then they start producing insulin. Yes?
So just follow up on the tolerance that you're generating. Is this peripheral tolerance, or do you have to continually give your antigen so you maintain tolerance, or is this something that is curative and we have some...
Very good question. Very good question. In all of these studies are being investigated. I can answer your question from our recent results on the hemophilia, where when they develop this tolerance, the inhibitory antibodies and IgE is completely undetectable levels. but after 6 or 7 months, the level starts slightly climbing up. So we were able to give additional dosage and then eliminate that. So we project that there may be, because this is a lifelong treatment, we anticipate probably treatment once in 6 months based on extrapolating from the mouse studies. So there may be a necessity for this for diabetes. We are now continuing this, to figure out, when. But interestingly enough, in diabetes, we never got this, autoimmune thing back. So I suppose that it may vary from the disorder to disorder.
Do you have plans to actually make something to try to get, or to specific targets and get central tolerance, and then it's not an issue in the future, or is it just luck that in diabetes it actually works any different than hemophilia?
Well, it's not, because you see, in reality, the two are not fully comparable, because in one case, the lack of production of clotting factors is gene, stop codons and all the other, several other things. Whereas here, in type 1 diabetes, there's an active immune attack to stop the production. So they're not really comparable, but I suppose that there will be a need for subsequent doses in the future. Okay, so I have 5 more minutes?
Yeah.
Okay. So this is, again, sticking with my earlier comment on interdisciplinary science. I've now talked to you about genome, genes, bioinformatics. I didn't have time to show you all your genome structures and so on, but then recombinant DNA production, expression, and immunology. But you could also add some more to your learning process, looking into environment, and go beyond the labs and look at other challenges. In this case, one of the, just as we have health challenge, we have energy challenge, and the production of fuels and fossil fuels. And one of the major challenges right now, why the ethanol that's made in the U.S. and in Brazil is sugarcane sugar. In the U.S., it's cornstarch. And so it takes food away from for the fuel purposes. And the best solution would be to use the entire biomass. If you use corn, just don't use that 1 foot of corn that we eat, but use the rest of the corn plant. If it is sugarcane, don't use the extracted sugar, but right now in Florida, the sugarcane, after the harvest season, they are burnt. So it's a major environmental challenge. But why is it then this technology is not being used? Because the enzymes that are needed to make this are insanely expensive. Some of them are $1 million. So you really need many of these enzyme cocktails, and you also need them to produce them cheaply. So we use, again, the chloroplast transformation system, and this paper also came out a couple of weeks ago, and we developed all what is called as core enzymes. Core enzymes needed to degrade the biomass, and from that, use that to digest the plant biomass itself. So we again here compared the fermentation system with the plant-derived enzyme system. As you can see, the plant-derived enzyme system works really well. So we made enzyme cocktails for pine wood. You can see from wood you can make sugar, and using the enzyme cocktails that we made. And these enzymes are anywhere between a few thousand to 100,000 times less expensive than the current enzymes. We also, in Florida, the citrus, our oranges don't look pretty, so we just squeeze them and make juice out of it, and then the rest of the peels are burned. So the projection of DOE is that we can make 500 million gallons of ethanol from citrus peel. So we developed the citrus peel, and one of the enzymes that we developed, actually the beta-glucosidase, which is one of the core enzymes, we have this manuscript in review, where it actually turned out to be even more fun than really producing the enzymes. Here, the beta-glucosidase acts on the conjugated hormones, glucoside conjugated hormones, separate this, and then the free hormone, the inactive hormones cannot function, but when they are released, then you can see the plants growing double in biomass. And then the glucoside themselves actually are insecticidal. And so here is the biomass data that you can see. So if you want to use biomass, then you really need biomass. And here is the enzyme that also offers you increase in the biomass. And then the released hormone increase the trichomes. So these are very typical of gibberellic acid. These are the control leaves. These are the transgenic leaves. And then we looked at the, you can see these glucosides are released in the glands, and the insects that feed on them, you can, when we stain these glucosides, sucrose esters in red, the insects become red. But as you can see, just a slide. Yeah, you could see that if you guys are working in the greenhouse, you know how challenging it is to control the whiteflies. And our transgenic plants can be touched by these whiteflies because they just get killed when they feed on this. So we not only increase the biomass, but we also, and we did all these insect bioassays, and almost 15-fold to 18-fold difference in the insect population control. And scientifically, did basic research, we found that these are glucose esters. We identified the glucosides. Our collaborators in Germany estimated all the GA, the pathway, and then helped us to develop the hypothesis how this worked. Basically, the GA is hydrolyzed, so we could look at the precursors of GA, we could see free GA, we could also see metabolic products of GA to confirm that we have now deconjugated this, and therefore we can really release the hormones, and then we could really be benefited by the biofuels. So Nature Biotech is one of the premier journals in this field. So they actually rank these inventions, and for the past 10 years, they ranked some 10 major inventions, and they ranked this chloroplast technology as one of the promising technologies for the future. Of course, they ranked it for the biopharmaceuticals and the vaccines industry, and of course, Nature Biotech has often featured us on their cover as well. So I'll conclude with this slide that if you guys, since Valentine's Day is around, have you guys seen a rose of this color? So that's a genetically modified rose. You can order it in Orlando. So, and so just to be creative, even in celebrating your Valentine's Day, to include science as well. And well, I guess I'll answer some more questions, and then thanks, Brent, again. Thanks for the invitation.
So what genetic modification is making that rose purple?
Oh, there's a lot of genes that you can change the colors. You can even make black rose. So you just go into the anthocyanin, pathway, and then tweak a gene here, there, and then you can get a whole lot of fun colors.
Dr. Daniell, this far as some of the vaccines, I know we've talked before a little bit, are any of those being taken to maybe start some clinical studies or testing?
Yeah, that's a good question. In the U.S., clinical studies cost $100 million, so far all the inventions have been funded by NIH, competitive grants. But from now on, I have to get investors cough up that $100 million. And especially for diabetes, when I tell them that we don't need to sell insulin, there are not too many volunteers. But I anticipate that some of these human clinical trials wouldn't, I know for sure, wouldn't cost $100 million if it's done outside the U.S. So I anticipate that some of the human data will be generated outside the U.S. and then come back to us for phase 2 or phase 3 trials.
Well, it seems like some of the vaccine, especially, there's a lot of potential there, especially in other countries, maybe not, that are affected by malaria or some of these diseases.
Absolutely. For malaria, for example, I'm hoping that the Gates Foundation may fund the clinical trial for malaria and cholera. They're very interested in that. And so I'm hoping that, some of these, hopefully, this year and next year, we may have, once we have the first human data, then we will have more.
Yeah, then it'll be easier, getting that started. There's some more questions back there.
Yes.
So your malaria studies, what antigens or what stage are you at? Yeah, that's really hard to market for vaccine. And immune response.
Yeah, that's why we use multiple vaccine antigens rather than relying on one. We use AMA1, MSP. In many of these vaccines, we have, even for plague, we use more than one capsular protein. So it's known to change, but if you use the current strategy is to use multiple antigens to try and do that. And one of the challenges in doing the malarial studies is that we have developed the human malarial vaccine antigen, and this is true with insulin also. We did the human insulin in mice. Thankfully, we were able to use that. But in case of malaria, rodent malaria is different from human malaria. So we are finding it very difficult to have an ideal, we have modified the falciparum, to infect rodents. An Australian group did that, we tried to use that. So there are some challenges. So one of the ideas right now is that, do small group of studies in countries where people are, dying in large numbers, and get the data directly from humans rather than in model systems, which are really not a real model system.
If I can just interject a comment, one of the things that Dr. Daniell improved on, and one of the advantages of using chloroplast is you can introduce multiple genes. I mean, when you're talking about making transgenic animals or plants, the technology for introducing single genes has been around for a while, but getting multiple genes and getting them all expressed at the levels you need can be a real challenge. And chloroplast, part of it, chloroplasts are very bacterial-like in nature, and they have operons and things. So developing that ability in chloroplasts has been a big plus. So when he talks about expressing multiple proteins or multiple antigens, it's very doable, but a lot of that's been from other developments and publications from his lab in the past.
Yeah, that's a very important point. Thanks, Brett. Yeah, the multi-gene engineering we developed several years ago, so that made all these studies feasible, because none of the examples that I gave you, even proinsulin, A, B, and C, and then I fused with CTB. There are 4 genes we're talking about. So in many of these, we have used 4, 5, 6 genes, and thankfully we are able to express them in equal ratios, so that when that is necessary. Multi-gene engineering, and also a lot of these basic concepts I will talk about today regulatory sequences that work in one species doesn't work in another one, like lettuce doesn't work in tobacco, tobacco doesn't work in lettuce, and so on. So a lot of basic conceptual molecular biology work has gone into this.
Other question over here? Or if you want to come down or something.